Hello and welcome to this video on test cross Mendelian inheritance. In this video, I will present the definition, methodology, interpretation of finding, and the significance of test cross. I will present the overview of the test cross. From Mendel's monohybrid cross experiments, he predicted using the law of probability that in the F1 generation all plants were tall or dominant phenotypes and heterozygous. In F2 generation, 75% of the plants were tall or dominant phenotype, of which one-third were homozygous and two-thirds heterozygous. 25% of the F2 generation plants were dwarf or recessive phenotype and homozygous. Since the recessive phenotype or dwarf phenotype appears in homozygous or pure condition, there is no need to determine the genotype of the recessive phenotype plant. However, the tall or the dominant phenotype plants could be homozygous and heterozygous. Therefore, Mendel developed an easy test which is known as test cross or back cross to determine the genotype of the plants showing the dominant phenotype. In the next slide, I will present the methodology of the test cross. Let me explain the methodology which he used in test cross. As usual, the method was elegant, simple and straightforward, in which he crossed a test plant with unknown genotype but dominant phenotype with a pure recessive control plant. Recessive is always pure, so this was homozygous in nature, and just using the offspring phenotype, he was able to predict the genotype of the test plant accurately. There are two types of test crosses. One is monotrait test cross and second is two-trait test cross. I will focus on the monotrait test cross in this video. So let me discuss monotrait test cross or monohybrid test cross and examine how the genotype of the test plant will affect the phenotypes of the offspring plant. So in this case, recessive dwarf plants which is homozygous was crossed with the test plant having, let's say, the genotype is heterozygous. So genotype for the recessive dwarf is homozygous, so small t, and the genotype for the test plant, let's assume it is heterozygous, make gametes from both parents. From this parent, gamete genotype will be small t. From this parent, it will be capital T small t. So make two small square and put the corresponding combination in the square and predict the phenotype. So this will be dominant phenotype or tall, this will be recessive phenotype or dwarf, and therefore a heterozygous test plant will produce 50% tall dominant and 50% recessive or dwarf phenotype plants. Let us examine how the test plant with homozygous genotype capital T will affect the genotype of the offspring plants. So genotype of the recessive dwarf, which is control homozygous, will be small t, and for the tall test plant, it will be capital T homozygous. So gametes from both parents will be one type, small t from here and capital T from here. Make a small square and put the combination here, predict the phenotype of the offspring plant, which is dominant phenotype. Therefore, a homozygous test plant will produce 100% offspring with dominant phenotype. So let me go over with the significance of test cross. The first is it helps in determining the genotype of an organism showing a dominant phenotype. It also confirms the Mendelian inheritance patterns and validated the genetic predictions of Mendel's experiment. In addition, it helps the breeders to select a true breeding lines of plants and animals so that they can grow good quality food and fodder. And finally, the test is useful in research and agriculture to maintain or create desired traits of plant and animal. Thank you very much for watching this video. Please like it, share it and subscribe it. I will be posting more videos on Mendelian inheritance. So watch out and take advantage. Until next time, you take care. Bye bye.